Hello again, Nicole Mashburn, and now we're going to talk about the events at the neuromuscular junction. So to this point you should have learned about the neuron and how to generate an action potential. Well, the action potential is now going to go all the way down the neuron into the muscle, and we're going to talk about what happens where the neuron and the muscle come together. So when you're going to initiate a response, so let's say you see a glass of water. Your eyes take that water as a sensory input to your brain, and your brain says, wow, that water looks great, I would love to have some. And it sends out a motor output to the muscles in your arm to pick up that water. And so this is the way basically everything functions. If we sense our environment, and then our brain makes a decision to respond. And so what we want to talk about today in particular is what happens when that motor output, that decision that's coming down from the brain, how does it get from the nervous system to the muscular system? So how does that change from an electrical stimulus from the brain turn into a physical contraction of the muscle? So just to review, this is a neuron, okay? So we've got our brain up here, we're sending down the signal, it's going to go to a neuron in the spinal cord. We're going to assume this is going to be the neuron in our arm, okay? So this is the output. This neuron is going to generate an action potential and send that action potential all the way to the end, to the axon terminals, and then there will be a muscle at this end. Okay, so what happens at the neuromuscular junction? Okay, so we have the neuromuscular junction. And the neuromuscular junction is going to be about halfway down the length of a muscle fiber or a muscle cell. And remember, this is where your synapse is. So you have an axon terminal, you have a muscle fiber, and between the two you have a synaptic cleft. It's filled with gel, which allows your neurotransmitter to float across from the neuron to the muscle to cause an effect. So in your axon terminal, you have synaptic vesicles that contain your neurotransmitter. And in this case, the neurotransmitter is called acetylcholine. Now, a neurotransmitter is something that's made by the nervous system, okay, that can be released by the nervous system, and it will go out and bind to a receptor and cause an effect. Okay, so remember we had those ligand-gated channels. Earlier I talked about ligand-gated channels. So acetylcholine is going to bind to a protein as a channel and cause that channel to open and allow sodium to move. Okay, so this is the ligand. This is the thing that will bind a channel and cause a change. All right, so where does this acetylcholine bind? On the sarcolemma. So remember the muscle fiber, the cell membrane, the plasma membrane of a muscle cell. It's called a sarcolemma, and it has receptors for this acetylcholine. Okay, so the neuromuscular junction is where the axon terminal and the muscle fiber come together. Your axon terminal contains your acetylcholine. Your muscle fiber has the receptors for the acetylcholine. So we want to release the acetylcholine and allow it to attach to the acetylcholine receptor on the muscle fiber, and something's going to happen. Stay tuned. So here we are at the neuromuscular junction. So we have our neuron and our muscle cell, and this is where they come together. Now they don't physically touch. Where the neuron and the muscle come together is called a synapse, but there's actually a space called a synaptic cleft. So they don't physically touch each other. There's actually a space between the neuron and the muscle fiber, muscle cell. So what happens is we've got this action potential coming down the end of the axon to this axon terminal, and we need to do something in order to get this signal from the nerve over to the muscle. Well, because they don't actually touch, we have to basically cause something to be released from the axon terminal that can then travel to the muscle to cause something else to happen. Okay, so we're going to take an electrical event, bring it down the axon to the axon terminal, cause a chemical to be released, have that chemical go to the muscle and cause something to happen on the muscle side. So at the synapse, your action potential will arrive at the end of the axon terminal, and it will change the voltage of the membrane and open up voltage-gated calcium channels, allowing calcium to flow into the axon terminal. Well, this flood of calcium into the axon terminal is the signal to release these vesicles. And these vesicles are storage places for a neurotransmitter called acetylcholine. So once the action potential travels down to the end of the axon terminal, the voltage changes, opening up these calcium channels. Calcium floods into the axon terminal, causing these vesicles to exocytose, remember, to release their contents out into the synapse. So they're going to release acetylcholine, which is a neurotransmitter, out into the synapse. Okay, so just review. At the neuromuscular junction, your action potential, nerve impulse, comes all the way down to the axon terminal, causes a voltage change so that which ion? Calcium will enter into the axon terminal. That will cause those acetylcholine containing vesicles to basically exocytose or send out the acetylcholine across the synaptic cleft. It will diffuse across that cleft, bind on its receptors, and when it binds its receptors, remember that's a ligand-gated receptor, it's going to cause a flow of something, possibly sodium. And remember, if sodium and potassium start flowing, what happens? You get a charge thing again. So we're going to basically start another action potential. So we're going to take an electrical event, turn it into a chemical event, back into an electrical event, and then eventually into a contraction. Okay, so here are all the steps of the events occurring at the neuromuscular junction, and you have this in your textbook as well. So step 1, the action potential arrives at the axon terminal, causing a voltage change, opening up calcium channels. Calcium floods into the axon terminal, causing the release of acetylcholine by exocytosis into the synaptic cleft. Acetylcholine, which is the neurotransmitter, will diffuse across the cleft and bind to its receptor. Okay? When acetylcholine binds to the receptor, this is a ligand-gated channel. That means once it's been bound by the acetylcholine, by the ligand, it opens. And the opening of this channel allows the movement of sodium and potassium across the membrane. And this movement of sodium and potassium across the membrane causes a membrane charge change on the muscle side, okay? And this will occur and continue to occur as long as acetylcholine is out in the synapse and can bind the receptor. So between muscle contractions, to stop a muscle contraction, you've got to get rid of the acetylcholine, and that can either happen because acetylcholine will just simply diffuse away, or it can be broken down by an enzyme called acetylcholinesterase. Okay, so we have an action potential coming down from the neuron, causing calcium to enter into the axon terminal. This releases acetylcholine into the synaptic cleft. Acetylcholine binds a receptor. This receptor starts to allow sodium and potassium to change and to move across the membrane, which is going to start making a charge change, which is going to lead to an action potential. And then that will occur as long as acetylcholine is in the synapse. We get rid of the acetylcholine, this will stop. So the next thing we want to talk about is what happens once the sodium and potassium channel has opened. What's going to happen? What happens to the muscle side? All we've done is we've taken an electrical event, turned it into a chemical event, the acetylcholine, and now we're back to an electrical event. We're basically going to spread this electrical event, this action potential, all the way across the muscle. The muscle still hasn't contracted yet. We've got to basically get this signal all the way down the muscle cell so then it can contract. Remember, a muscle cell is really long. The one in your arm, if it's the biceps brachii, goes from your shoulder all the way to your forearm. So a really long muscle. So we've got to get the entire muscle electrically activated so that we can then contract it as a unit. So how do we generate the action potential on the muscle side? We have a local depolarization. That acetylcholine, once it binds that channel, that ligand-gated channel, that causes the simultaneous diffusion of sodium into the cell and potassium out of the cell. More sodium diffuses in, so the interior becomes less negative. it depolarizes. So that's a local depolarization. So acetylcholine binds that ligand-gated channel. More sodium enters, the potassium leaves. The cell membrane at that point starts to depolarize. So let's see what that looks like. So here comes the acetylcholine. It binds the channel. Sodium rushes into the cell, the muscle cell. Potassium leaves the muscle cell. This causes it to become slightly positive, to depolarize. Well, this starts a depolarization wave that will spread in both directions down the muscle sarcolemma. And as this wave of depolarization moves down the cell membrane, more sodium channels open, allowing more depolarization. And then, just like in the neuron, you'll have some potassium channels will then open, and you'll get repolarization. And then you'll just see the action potential will propagate or move all along the sarcolemma of the muscle cell. So you took the action potential down the axon terminal. It couldn't jump the cleft. It can't just physically jump over. So we had to cause acetylcholine to be released. It has to bind the channel receptor on the other side, on the muscle cell. This starts a new wave of depolarization and action potentials on the muscle side. So just like in the neuron, in the muscle cell, the sarcolemma has a wave of depolarization and repolarization, or action potential, spreading across that sarcolemma. The depolarization is due to sodium entry, and the repolarization is due to potassium exit. Okay, just like in the neuron. So this action potential that's moving across the cell membrane of the muscle will occur as long as there's acetylcholine in the synaptic cleft. So we need to destroy acetylcholine or remove acetylcholine from the synapse, and we do that using either diffusion, it just floats away, or acetylcholinesterase, which is an enzyme that will break down acetylcholine. So the rest of this video is going to be an animation of events at the neuromuscular junction. So you'll watch this, and in my next video, we'll talk about what happens next. So once this membrane potential, this action potential has moved all the way across the muscle cell, the next step is to actually physically cause the muscle to contract. So watch this video, and I'll see you in the next video, and we'll talk more about the next steps of contraction.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open, and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.